Okay, so today we're going to be talking about diffusion, right? Specifically diffusion across a membrane. Now, why is this important? There are two questions that you want to keep in mind to think about the importance of diffusion, because this all is stuff that you have to learn before you get into the pharmacology. First thing you want to ask yourself is, how do drugs entering the cell have anything to do with diffusion across a membrane? So, drugs entering the cell, right? The second thing that you want to ask yourself is, what types of diseases are associated with poor diffusion, right? So question number 2 is diseases associated with poor diffusion. If you keep both of those in mind, then you'll kind of get an idea of what you're supposed to be looking for throughout the video. Okay, so what is a membrane, right? So we have a bunch of membrane-bound organelles inside of a cell. Specifically, what a membrane does is that it separates or acts as a barrier for ions, right? So let's say I have my cell, right? And this is my cell membrane. On the inside and on the outside, we're going to have different concentrations of different ions, like sodium, potassium, chlorine, or chloride ions, all these different things, right? So just to give you an idea, on the outside of the cell, you're going to have more sodium. Okay, so sodium is going to be high on the outside. You're going to have also more chloride. You're going to have more potassium on the inside of the cell, right? And you're also going to have more hydrogen ions on the inside. What I mean by that is you're going to have a lower pH on the inside. It's going to be slightly more basic on the outside. Not that much, right? In terms of specific organelles inside the cell, if you were discussing, for example, the mitochondria, right? So let's say this is our mitochondria. Our mitochondria, something to keep in mind about mitochondria is that it's going to have much, much lower voltage, right? So it's going to have a negative voltage, okay? And then if we're dealing with lysosomes, which we know lysosomes are there, they have basically things that break down different proteins and stuff like that. That's going to have a lower pH, right? So lysosomes are going to have an even lower pH. So all those to keep in mind. Again, all of these membranes allow for that difference to occur. You need the membrane for that negative voltage. You need that membrane for a lower pH. Something to also keep in mind, just so that you can remember the sodium and potassium on the inside and outside. Remember, if you have a sodium-potassium pump, right, NaK pump, you are pumping sodium out and you are pumping potassium in, right? And that's going against the gradient, right? So pumps go against the gradient, and Na out, potassium in, right? Okay. So in terms of diffusion, what exactly is happening? When we're dealing with diffusion, we're dealing with a specific particle that is moving randomly around its origin point. So you have one particle, right? And it's moving around its origin point a certain distance, okay? As more time passes, so more time passes, diffusion is going to be farther. So this particle moves farther from its origin point. If you remember that, it's pretty simple. The way that I like to remember diffusion is by keeping in mind Febreze, right? right? If you think of diffusion as a Febreze spray, you'll be able to remember all the different rules associated with diffusion. So just think of diffusion. Whenever we're thinking about it, Febreze is our example, okay? So we're gonna get into certain rules or factors that affect diffusion. The first rule that we deal with when it comes to diffusion is Fick's first law of diffusion, right? So Fick's first law. So in Fick's first law, we have an equation. It is J is equal to negative D delta C times delta C over delta X. This might seem like a lot, but it really makes a lot of sense, okay? And really the point is not to remember the equation, but the rules. So J is simply flux, okay? D is the diffusion coefficient. And this delta C over delta X is representing the concentration gradient, okay? Concentration gradient. Okay, so what is flux? Flux, diffusive flux, is basically the amount of a substance or the number of moles per unit area per unit time, right? So number of moles over area times time, okay? So again, if we're thinking about that Febreze, right, we're basically saying that something with a higher flux means that we're having a lot of that concentration, a lot of moles in a specific area at a specific time. So if I'm having a lot of Febreze that I just sprayed in one area, it's gonna have a higher flux and it's going to move from that higher concentration to that lower concentration. So that's all that Fick's first law is basically telling us. All it's telling us is that diffusive flux goes from higher concentration to low concentration. And you know this rule, that high concentration to low concentration rule, okay? That's it. There's nothing else to it really, okay? And so then flux is just directly proportional to the magnitude of the concentration gradient. That's what this is telling us, right? So flux is proportional to that concentration gradient. You have a higher concentration gradient, you're gonna have a higher flux, right? Or a higher movement, right? That's it, okay? The next thing that we want to consider when we're talking about diffusion is permeability, right? So what is permeability? Permeability just means how well it's going through the membrane, right? So there are certain factors of the membrane that actually affect how well it's going to diffuse, right? Because diffusion is movement, right? So diffusion equals movement. And permeability is just how well it's going to go through the membrane. So will it go through membrane well? And what do you think are the factors that affect permeability? They're all common sense factors, okay? So in terms of permeability, the things that affect it are things like membrane thickness, partition coefficient, and the diffusion coefficient, right? So if I were to write this into equation, it would be P is equal to dm Kp over delta X. This is super easy. So just because there's a bunch of, letters, let's not get confused or anything. So P is permeability, right? That's it, that's just permeability. And what affects permeability, okay? The diffusion coefficient, right? So if diffusion is going well, then, you're going to be moving through the membrane well, okay? The partition coefficient. So what is the partition coefficient? So partition coefficient is basically talking about is something hydrophobic. And I'll tell you what that exactly means. Right, so as we know, membranes are hydrophobic, right? They're made of phospholipid bilayers, right? At least the cell membrane is made by a phospholipid bilayer. So Kp is just sea oil over seawater, right? So if I put a drug, for example, inside a tube, right? If I had a tube and I had it filled with oil, this is my oil, and then I had it filled with water. All right, this is my water, right? The question is, how much of it will be in the oil and how much of it will be in the water, right? If I have more in the oil, that's that sea oil, right? If I have more in the water, that's that seawater. So my higher Kp equals more in the oil. More drug in the oil, right? And all that means is that my drug is just much more hydrophobic, okay? And high lipid solubility or high hydro, if it's more hydrophobic, means high membrane permeability. So that's really that rule, okay? So high lipid solubility equals high membrane permeability, or just high permeability, right? Okay. Second thing that we want to consider is delta X, right? So if you remember, if we look up here, delta X is on the bottom, right? So as the thickness, delta X is just equal thickness of the membrane, okay? As thickness increases, so increase delta X, permeability decreases, right? Think about it. If I have a thicker wall to get through, is it going to be harder to get through or easier? It's going to be harder, right? That's it. That's all there is. So things to consider, again, okay, are basically permeability increases with lipid solubility, right? And decreases with membrane thickness, right? Another two factors. So first we mentioned, right? So let's write these down so we don't forget them. So the first thing that we talked about was lipid solubility, right? The second thing that we talked about was membrane thickness. Okay. The third thing that we're going to talk about is molecular size. Okay. So if I have specifically, if the particle is smaller, right, then it's going to be able to move farther. Okay. That's the rule with diffusion, right? So let's say, for example, I have something that isn't very lipid soluble, but it's a smaller size, then I'm going to have an increased permeability. It's going to be more likely to be able to get through my cell membrane. So permeability increases with increased lipid solubility, lower membrane thickness, lower size of particle, and when it comes to the polar charge or like the ionic charge, that's super hydrophilic, right? Because those associate with water. Ions associate with water. So permeability increases when charge decreases, okay? So those are the four factors that affect permeability. So what does all of this have to do with drugs, right? If we want to get to basically the real medical relevance of all of this stuff, okay? So when it comes to drugs, some drugs, for example, might not be that hydrophilic. So what are we going to do? How are we going to get it inside the cell membrane? Okay, there are two ways to get drugs in. Okay, the first way is to add a hydrophobic portion to the drug so that it can enter into the cell, right? So let's say I have a drug and it's not that hydrophobic. Okay, so it's like kind of hydrophilic, but then I add this little portion right here and it's very hydrophobic. So now this will enter the cell membrane, okay? And then that piece can then be broken off, right? So usually what it is, it's usually an ester linkage, and the ester linkage just breaks off and this just goes out. It doesn't matter what happens to this, and then drug works, right? The second way, so this is the first way. The second way that we have stuff is that we add a specific portion that actually targets a transporter or a receptor, right? So for example, if I have a drug, again, like this is my drug, right? And again, it's a little bit hydrophilic, you know, it's not gonna be able to get in. But then I add this portion right here, and this is actually a ligand that attaches to a receptor on the membrane. So this is my membrane. And then let's say this is my receptor. So it matches this right here. That can get in, and again, the drug is able to work inside, right? Okay, it works. So those are those two ways. Okay, the second question that remember I asked you was, what are some diseases that are associated with basically drugs that, what are some diseases that are associated with poor exchange through a membrane or poor permeability through the membrane, right? So let's think about somewhere where a membrane is very important, the lungs, right? Specifically, the alveoli. So gas exchange occurs through the alveolar capillary membrane, right? If you have a higher thickness, what do we say happens when you have a higher thickness? You have poor permeability, right? So remember, increased thickness, lower permeability. So there is some diseases that basically you have a thicker membrane in the alveolar capillary membrane, right? So if I have my alveoli, right, like it's that little air sac, right, and it's supposed to have some type of diffusion of the O2, right? The O2 is supposed to be released and then goes into the blood, right? What happens is that if I have a thicker membrane, right, if my membrane instead is like this, my problem will be that I'll have a lower O2 diffusion, right? This is simply called, so this is for the alveolar, right? This is called fibrotic lung disease. So fibrotic lung disease just means increased thickness, okay? The second disease that we're going to be talking about is, well, it's not really a disease, it's more of a condition, is a pulmonary edema, right? So let's say you have fluid in the lungs, right? Fluid in lungs, okay? This is called a pulmonary edema. What's occurring there is that because of the fluid in the interstitial space, there is a greater distance that must that the diffusion must travel through, right? So instead of, for example, here is the alveoli and then here is the blood, right? It's more like this. So this increased distance, we talked about how diffusion occurs faster through smaller distances, right? So because of this greater distance, okay, we're again having a decreased PO2, okay? So greater distance for particles to travel. Okay. The last thing that we're going to just talk about very quickly is a little bit more about drugs, okay? So in general, keep in mind that when we're discussing weak acids and weak bases, so I know this is like where acids and bases coming from. The thing is that drugs are usually in the form of weak acids or weak bases, okay? So let's say I have a weak acid, okay? Is a weak acid, A negative, going to be able to enter into the cell membrane? No, probably not. Why? Because it's an anion right now, okay? So instead, the way that it needs to enter is by adding this proton, okay? And then entering as HA, okay? This is much more likely to be able to enter the cell. Same thing with a weak base. Let's say I have a weak base. The equation is H plus B is equal to BH plus. Which one is going to be able to enter? Is it the BH plus or the B? The B. Okay, the B is going to be able to enter. Why? Because it is not charged. So the non-charged form is more likely to be able to enter. So keep in mind that that's the same with drugs, okay? Other things to consider about drugs, okay, is that oral drugs are mostly absorbed across epithelial cells in the GI tract. So oral drugs, GI tract absorption, right? GI absorption. Okay, so in terms of that, things that will affect that is the surface area available for diffusion, right? So greater surface area means that more diffusion can occur. Okay, so surface area. Blood perfusion rates. So if blood is flowing slower, then that can impair the concentration gradient. Think about it. If I have blood moving very quickly, then there won't be a lot of this specific drug in this area so that I can move faster, right? So this is if I'm moving quickly. But let's say I'm moving very slowly, okay? Then there's going to be less diffusion into the blood, okay? So blood perfusion. And then the last thing we mentioned before is delta X, which is just thickness, all right? Thickness of the membrane. Obviously, increased thickness decreases permeability. All right, that's everything, guys. Thanks so much for listening.